Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you another really awesome guest involved in creating a better tomorrow for so many people out there. Today, we have the honor of being joined by Dr. Drew Weissman, 2023 Nobel laureate in medicine, Roberts Family Professor in Vaccine Research, and Director of the Penn Institute for RNA Innovation. He also serves as professor in the Department of Medicine and Director of Vaccine Research in the Infectious Disease Division at the Perelman School of Medicine down the street at University of Pennsylvania. And Dr. Weissman is a physician scientist, a pioneer in the science of immunology, making major contributions to the field. Obviously, most recently, he and his colleagues and the discovery of these novel nucleoside-modified mRNA platform, which obviously served the basis for the COVID-19 mRNA vaccines that many of us have taken, but also really serves as an important technology set for the burgeoning research involved in targeted therapeutics for some of the world's most devastating diseases, both communicable and non-communicable. And Dr. Weissman and his TAB team continue to pioneer the basic research in this space. Projects that his team is currently working on include, among other things, pan coronavirus vaccines, universal flu vaccines, herpes vaccine, as well as collaborations with other Penn colleagues in the area of cancer therapeutics. Prior to joining Penn, Dr. Weissman was a fellow at NIH studying HIV in the immunoregulation lab with Dr. Anthony Fauci. Did his bachelor's degree and master's degree at Brandeis in biochemistry and enzymology, then moved on to do his medical degree and PhD work in immunology and microbiology at Boston University, and completed his residency at Beth Israel Hospital. We're honored to have him. A lot of interesting topics to get into today. Dr. Drew Weissman, thank you so much for taking the time out of your schedule to come talk to us for a little bit.
Thank you very much. I'm honored to be here.
It's an honor having you. I would love to start things off going back to 1987, because it's at this point you publish your PhD, regulation of B lymphocytes with reagents that cross-link surface immunoglobulins, and here begins a theme that we'll see over and over throughout your career. I'm simplifying things here a bit, but here is a type of cell we're interested in. We want it to be stimulated, to activate for a certain function. Certain functions we don't want it to have, and we all want that to happen in the right location. What you define, I'll take some of your words out of your papers: the multiphasic and multifactorial immunopathogenic mechanisms of immunology. Take us back to 1987. What got you started in this particular domain of research?
Yeah, so it's a long time ago to remember specifics. I became interested in science and math and engineering when I was a little kid. But my parents used to joke that nothing in the house worked because I would go around and take everything apart to learn how it worked. So no doorknob worked, the toasters never worked. And that's how I started. When I was in college, I spent my summers doing basic science research at labs in Boston. So that exposed me to basic science. And I decided to actually do a master's degree while at Brandeis to get a better exposure. When I went to MD PhD training, I had fallen in love with immunology from my summers doing research. And moved into Anne Marshak-Rothstein's lab at BU and had a wonderful experience doing research and knew that that's what I wanted to do for the rest of my life.
And at that point, well, a couple of years later, after doing your PhD, you moved to NIH, into the immunoregulation labs with Dr. Fauci, and here's where you start working on this extremely important cell, the dendritic cells, a very potent antigen-presenting cell. Again, looking at what makes these cells mature, what makes them not mature, and all the cytokines and chemokines. Again, this multiphasic immunopathology. Talk about the 1990s, if you would, at NIH, because we've had Dr. Fauci on a couple of weeks ago. Dr. Diefenbach was on a few months prior, talking about the sort of 2023, where we've come with vaccines and biologics and HIV. Obviously, things were a little tougher back then as you were exploring. Okay, what can we do with these dendritic cells at that time? But take us back to the 1990s and your work at NIH, if you would.
So when I started in Tony's lab, the main focus of the group I was in were cytokines.
Mm-hmm.
And it was known back then that cytokines drove HIV. We didn't understand a lot. HIV had been discovered a few years earlier, but we knew how to make it grow. And that was adding cytokines to cells. I had an interest in moving in vivo, moving to primary cells instead of cell lines. And that's how I became interested in dendritic cells. They had just been described as a new antigen-presenting cell, and the data was suggesting they are the most potent antigen-presenting cell. So I thought that they must have a role in HIV and started to study them and studied how to make them, how to infect them, how they transfer virus, how they become activated, and what they do. Tony gave me free rein to do these studies, and we generated some great findings that I took with me to Penn.
And you arrive at Penn in the late 90s, and then it's at this point that I'll refer to as sort of your Reese's peanut butter cup moment, with Dr. Carrico, who is jointly awarded the Nobel Prize with you. You have your dendritic cells, obviously working on how to optimize them, activate them, learn more about them. She has her mRNA. And I believe it's in 2000 when you come together and you publish your first paper on HIV and how to transfect these dendritic cells properly. But this journey sort of begins on ultimately not just how you can modify the cells, how you can incorporate the mRNA, but then all these other components, how you design the proper lipid nanoparticles, how you prepare this cell to activate not just as an antigen, but as an adjuvant at the same time. Take us to sort of the link up with Dr. Carrico and a little bit of sort of the journey on everything that led up to what we have today in terms of the COVID vaccines, and then we'll be getting into some sub themes of that.
So Katie and I met. The story is true over a Xerox machine. Mm-hmm. Back then, the papers weren't on the internet. You couldn't read journal articles on the internet. The only way you could read them was to photocopy the printed journal. So every time the journal would come out, and there were hundreds of them, both Katie and I would fight over the copy machine to copy their articles in order to read them. And we started talking, and Katie talked about her prior almost 10 years working on RNA, where she was having great success in her research, but not in her career. She couldn't get grants. She couldn't get papers published. She was concentrating on neurologic uses of RNA. My interest when I came over from NIH was to use dendritic cells as a vaccine platform. They're the most potent antigen presenting cell. So what that means is they start all new immune reactions. And a vaccine is a new immune reaction. So my idea was that you load dendritic cells and you have to figure out what's the best way to do that. So I had protein and peptides and DNA and viruses and other methods. I didn't have RNA. I didn't have access to RNA. And that's where I met Katie on the Xerox machine. And she was telling me about all the different kinds of RNA she made. So in 87, when I started at Penn, we started working together. From there, it was about 8 years before we figured out nucleoside modification.
Mhm.
And during that time, we made a lot of interesting findings and we published a lot. We identified that RNA was inflammatory and why. We found the receptors that mRNA bound to and signaled through. And that led us to nucleoside modification as a way to avoid that inflammatory activity.
Mhm.
That turned out to be incredibly important for two reasons. The first is now you can give RNA to an animal and ultimately a person safely. You won't make them sick. And even more important, we increase the amount of protein made off of the RNA by upwards of 1,000-fold in an animal and ultimately in a person. So prior to that, RNA had essentially failed. It was in clinical trials for cancer vaccines. They would take dendritic cells out of a person, pulse them with mRNA, and give them back. They made immune responses, but it did no good. It didn't help with the cancer. So pharmaceuticals and biotechs essentially gave up on RNA.
Yeah.
They said, this didn't work. It doesn't make enough protein. It's too inflammatory. There's no reason to do this. And they all gave up. And there was maybe one biotech company that was still doing RNA work. All the pharmaceuticals gave up. Everybody else gave up. And RNA became a bit of a wasteland that people weren't interested in.
Yeah.
But Katie and I started working together, and we generated interesting data, and the data kept us going. But I think most importantly, the potential of RNA is what really kept us going.
Mhm.
We didn't see it as a treatment for one disease. We thought it had potential for many different vaccines, both infectious disease, cancer, and other. Thought that it could be used for gene therapy, for protein replacement, for protein therapeutics. We would make lists of all the potential diseases that RNA could be used for, and the list just went on for pages and pages. So that kept us in the field and kept us working, even though we couldn't get grants and we had trouble getting our papers published.
Mhm. Mhm. And yeah, I mean, if once again, if everybody listening and watching, if you go to Dr. Weissman's PubMed um, uh, at the NIH, I mean, it is an extensive array of communicable and non-communicable diseases that you've been publishing on as of late related to the technology. And I thought we could divide these up because clearly there's a lot going on, as you were just mentioning. When we look at sort of just so I'll call the traditional diseases. I mean, you published on the flu, on herpes, on CMV, and again, these are unlike the flu, obviously respiratory, but some of these viruses have different tropisms and so forth, the nervous system, mucosa, and all that. Talk a little bit about sort of what goes into your sort of daily decision process because they're all very different. Clearly, there's a unifying platform here. But one of the very interesting things in your Nobel presentation was the discussion about the multi-antigen nature of what you're doing, and the fact that aside from being able to study more than one sort of stimulating agent, that the past issues with what's, I guess, called antigenic competition doesn't seem to be arising with some of these technologies. And then that's, I think, another major benefit of what you're doing compared to older school approaches. Say a few words about that as well. I think it's extremely important.
Yeah, so when we started studying HIV was, of course, first, because I
Right.
was an HIV researcher. But we branched out into influenza, into herpes, into other diseases. Probably the linking nature to all of them were, these are diseases that other vaccines had either failed or had done poorly on. So no HIV vaccines still for 30 years. Influenza was around, it's less than 50% effective. Herpes had failed multiple times, malaria had failed, TB had failed. So we chose diseases where other vaccines had failed to see, would an RNA LNP vaccine work better? Along the way, we knew, and we had been adding multiple RNAs in both therapeutics and vaccines. And we discovered that we could add, we've added 20 so far for a
Yep.
couple of different vaccines. And what we saw that you don't see with proteins or inactivated viruses or other platforms, there was no antigenic dominance. One antigen didn't win out and block all the others. With 20 HA influenza antigens, we got the same response to all 20. With 19 tick salivary antigens, we got powerful responses to many of them. So we think we know why. It's because the immune response is so powerful, it's so big to RNA, which is why you get such high titers of antibodies. But it also gives you responses to many different. That will allow you to add many antigens into a vaccine. I joke sometimes when I give talks, and there's pharmaceutical execs there that I want to piss off a little. I'll tell them, someday your child could go into the pediatrician at 1 month, 6 months, a year, and 2 years and get a single injection of 20 RNAs that covers every antigen.
Yeah.
So instead of bringing your kid every month for the first 2 years of their life and then every 3 months after that, we could give a single injection that would give every childhood vaccine at the same time. And it would probably work well. It would probably work better. It'll never happen for a variety of reasons, but I like to think about it.
Sure. I can see why that would make people mad in the audience. What are you going to do? Dr. Wilson, aside from you just, went on a decent list of all the sort of viral and non-viral conditions that we're familiar with. You've also been publishing on some more obscure stuff. So, I mean, I saw a paper on something known as Crimean Congo hemorrhagic fever. You have a paper on Hendra, which is really, a Hendra soluble glycoprotein vaccine against Nipah. And we talked about Hendra actually with Lin Fa Wang down in Singapore last year, a pretty nasty virus there. But, I guess a lot of this coming back to this whole theme of emerging viruses that we should be concerned about, and sort of this One Health concept. Say a couple words about this as well, if you would, because clearly these are non-traditional things, but they're out there, and we should be thinking about whether it's human vaccines, or in the case of Hendra, where they went and vaccinated, all the horses back in the day, and stop that sort of One Health transmission. Talk about some of the applications per sort of the One Health team and the platform you're working with.
No, so to me, that's critical, but really for two reasons. One is many of those diseases are region specific.
Yeah.
You don't find them throughout the world, but you find them in people in specific areas. But most importantly, you find that those are diseases of animals, and many of these are food crop animals.
Yeah.
And if you look at how that works, if a chicken gets influenza, or if a pig gets Crimean Congo, or a sheep gets Kuru, or all of these diseases that are transmissible to man, when an animal food stock gets it, they kill millions of animals because the risk of the disease spreading further is greater than the cost of that animal stock. So it's important both for human health as well as food for people. Once it starts spreading through sheep or pigs, then it starts spreading more through humans. So you have to understand it's a big cycle, and you have to act everywhere to break that cycle.
Headed over to HIV for a moment, while we're on the communicable side of things. In January 2021, you published a paper, Safe and Effective In Vivo Targeting and Gene Editing in Hematopoietic Stem Cell Strategy for Accelerating Development. And last year, we profiled one of the CCR5 Delta 32 stem cell transplant cases. Clearly, that curative intervention, as it standardly it was done, not applicable for large parts of the population. But obviously, with your technologies and hematopoietic stem cells combined, that amongst the curative interventions possibly becomes much more, well, less expensive and a much larger targeted population. Can you say a little bit about applications with HSCs and your tools? HIV, but there could be other things involved there as well.
Yeah, so we have a very big program that's working on HIV cure. We have a separate program on HIV vaccines. But for the HIV cure, to me, the most important part is ability to use it throughout the world. As you say, you can't do bone marrow transplants on the entire world.
Right.
The cost, the mortality, the feasibility is impossible. But using mRNA LNPs delivered IV, that can be done anywhere. It's simply you thaw the vial, you inject it into a person, and if you design the therapeutics correctly, you may be able to cure HIV. Now that we're combining many different therapeutics. So one is, as you say, knocking out CCR5. That can be easily done. We've done it in mice and in macaques with high efficiency. But you need more than that. We've also been able to target resting CD4 positive T cells. These are the cells that HIV is latent in, that HIV hides in for essentially the entire lifespan of an individual. We can target those cells and deliver enzymes that either cut out or inactivate the proviral HIV and kill the latent reservoir. We can deliver broadly neutralizing monoclonal antibodies to the liver and produce high levels of neutralizing antibodies to be used as an immune effector. We can deliver HIV-specific CAR T cells, either the T cells or the bone marrow stem cells, and they're another powerful effector. That leaves one thing open, which is you have to activate the latent virus in the T cells in order to kill it.
Yeah.
If it's hiding in a T cell, the immune system doesn't know that. So you need a way of turning it on. What we call those latency reversal agents, LRAs. There have been a bunch have been in clinical trials. They've done nothing. They've done no good. And all of them are host directed. So what that means is it targets host cells and tries to activate the host cell to make the virus grow. We've come up with a new approach, which is to put the HIV protein TAT in CD4 targeted LNPs.
Okay.
TAT only works on the proviral DNA and specifically turns it on. So now we have a viral directed therapeutic that will turn on HIV and allow the CAR Ts and the bNAbs and the other effectors to clear the remaining infected cells out of a patient and potentially cure HIV. And all of it can be done with just an IV injection of RNA LNPs.
Extremely exciting. Yeah, extremely exciting times for in HIV. And we've come, again, you started when it was a death sentence, and to hear about these types of you know initiatives happening in 2024 where we're getting close to a cure is just it's very inspiring. So no, I'm excited to hear about that. Um, let's hop over to non-infectious conditions, because clearly you're doing a lot here as well. And again, thinking to your Nobel presentation, you said you talked about CAR T there, not just for cancer and the ability to present multiple antigens, in terms of cancer vaccine development, but a really, and as you say, you're going to piss off pharma with the vaccine stuff. You talked about this concept of internally generated CAR T via the LPS delivery systems. Here again, and we had Bruce Levine on a couple months ago. And CAR T, incredible technology, very expensive. You're coming with something here that sort of throws everything around, makes it a lot cheaper, and allows us to produce these CAR Ts internally. Talk a little bit about this. And for everyone that hasn't listened to Dr. Weissman's Nobel talk, go listen to it after you listen to our show.
Yeah, I mean, CAR T therapies are one of the most fascinating and most successful new cancer therapeutics. They work on liquid tumors, lymphomas, and leukemias. They can have success rates as high as 60 to 70% in patients that otherwise had a death sentence. The issue with them is they're made in a specialized lab.
Yeah.
They cost almost $500,000 a dose, and they can only be done where you have these specialized labs, which is mainly in the US and Europe. There's one site I know of in China. There's a few others scattered in high-income countries. There's nothing in low and middle-income countries. So what we wanted to do is we wanted to know, could we make CAR T cells in the body? And we were able to target T cells with mRNA LNPs. So we put a CAR in the mRNA. The RNA CAR encoded the CAR molecule. We targeted it to T cells, and when we injected this into mice that had cardiac fibrosis, and we gave them a CAR that got rid of fibrosis, we cured the mice. We fixed their hearts. We're looking at this going forward for both oncologic diseases, but probably more importantly for autoimmune diseases.
Okay.
There have already been clinical trials using CD19 CAR Ts for lupus. And these are lupus patients that have terrible disease that drugs can't treat. And when you give somebody a CAR, you've got about 9 months that they have no B cells. It takes 9 months for their B cells to come back. And you have to give them antibodies during that time. What was fascinating about these lupus patients is that when their immune systems came back, when their B cells came back, lupus didn't come back. it somehow reset their immune systems. How long it's going to last, I have no idea. Someday they might develop lupus again. But if we've bought them years off of with no disease, that's fantastic. Now, if you can do it in vivo with just an off-the-shelf RNA LNP at a cost that's a fraction of the ex vivo CAR Ts, now you've got a treatment that you can give around the world in settings where autoimmune diseases are not responding well to other treatments and are threatening people's lives.
Well, while on that topic, could you just say a few words about what you're doing in Thailand? Because I know you're passionate about translating these technologies to lower income countries. I know there was the work at Penn with the country Costa Rica and sort of translating some of the CAR T technology. But say a few words about what you're doing in Thailand, because I think this is, while we're on that theme, important that this is, much more than just what's happening here in Philly. But
Yeah, so I've worked in the field of equity my entire career. I started when I was a fellow at the NIH and first set up a lab in Thailand back then. My interest, so with Thailand, I've been working with the researchers at Chulalongkorn University for almost 10 years. And after, I knew Kiat, who is the head of their VRC. When we were talking, he expressed interest in having access to this new technology, and I was more than willing to help them set it up. So I helped him set up research infrastructure. I taught his people how to make RNA, how to design RNA, how to use it in vaccines. He has now a large group of researchers that are making vaccines for dengue, for
yep.
tularemia, for dust mites, and a variety of other diseases. When COVID-19 hit, in talking with them, we all came to the conclusion that any vaccine made in the West would take years before it ever was distributed in Southeast Asia or Sub-Saharan Africa or South America. And we saw from COVID, they gave tens of thousands of vaccines for hundreds of millions of people.
Mm-hmm.
Not very usable. But Kiat talked to the princess and to the government, and they were willing to fund making a COVID-19 vaccine in Thailand. That involved the basic science, and Kiat and I worked together to design a Thailand COVID-19 vaccine. But we also had to build a GMP production site. So GMP production is the quality of manufacture needed for human drugs and vaccines. So we built the GMP production site in Bangkok, and they have been making vaccines that Kiat's group develops for clinical trials. The idea is that they can now make their own vaccines, and they're making vaccines for local diseases, things that pharmaceutical companies are less interested because they're not widespread, they're not present in high-income countries. They won't make any money off of them. Now Kiat and his people can make vaccines that they need in Southeast Asia. The next time there's a coronavirus or influenza pandemic, they're set to make their own vaccines. I've done the same in Sub-Saharan Africa,
Mm-hmm.
in South America, in Eastern Europe. What we've built so far, 18 GMP sites around the world.
Really outstanding. Let's transition now to some of the other things you're working on at the lab, because again, you have quite a few really interesting areas that we've touched on the show, and I'd love to see sort of how sort of the nucleic acid Kant's topic, interacts with these different domains. So one, is the microbiome. Just last year, you published in February regulation of nucleic acid vaccine responses by the microbiome. And again, here we are, we're increased realization about these trillions of organisms that live on within us, made up of mRNA and other materials. What are you learning in terms of, how the microbiome affects these core technologies? And then, I guess the reverse, because I saw, it's interesting because I saw you published several years ago with Jack Stapleton. We had him on, talking about GB virus C and sort of some of the beneficial stuff that we're learning about the sort of the virome. And anything interesting in that direction, in the sense of how these technologies possibly one day, whether it's bacteriophage therapy or something else, it can potentially amplify the beneficial effects that we're learning about of our microbiome and viromes.
So what we've learned so far is that it's a two-way street.
Yeah.
That the microbiome alters the therapeutics, vaccines, drug treatments that we're giving to patients. And going blindly into a GI treatment without understanding what the effect the microbiome has on it runs us into trouble. We have patients who have chronic C. difficile infection. In the old days, we kept giving them antibiotics and they kept recurring. It wasn't until we figured out, oh, we screwed up the microbiome that we need to fix. Once we fixed the microbiome, then the C. diff disappeared. Just as well that vaccines that we deliver might alter the microbiome that could potentially lead to trouble down the road. So we've made a C. diff vaccine, and we've spent a lot of time being sure that this vaccine that alters the bacteria in the gut doesn't alter the microbiome, leading to difficulties somewhere else.
Got it.
So the crossovers between understanding the two are enormous. I'm fascinated by the virome research that's going on. All of the viruses that they're identifying in our GI tracts that we had no clue were there before. And most of them are just commensals that seem to grow and divide and disappear. Every so often you find some that turn out to be pathogens under certain conditions. If you immunosuppress somebody, they can suddenly appear. So the learning there is really enormous, and I think it's going to change how we do a lot of our therapy. We may determine somebody's microbiome before we select a treatment for Crohn's disease or for ulcerative colitis or for other chronic GI diseases.
The Polaris Dawn space mission is headed up, I believe, in April of this year. And one of the, it's partially SpaceX involvement here. And, one of the research programs within Polaris Dawn is to better study the effects of space flight and space radiation broadly on human health. I was really intrigued by a 2012 paper that you wrote entitled Effect of Solar Particle Event Radiation and Hind Limb Suspension on GI Tract Bacterial Translocation Immune Activation. Clearly, we're in this era of sort of the space race on steroids, the space economy. Not as much going on probably related to how all these things like radiation and microgravity and everything else affect us, but once we get up there, it's a pretty dangerous place. What are you learning about the microbiome in space? Where do we need to go with this research? Because clearly, as we send ourselves up there, we're sending up trillions of other organisms within us. Say a few words about that, if you would.
Yeah, so the work that I did over 10 years ago,
Yeah.
it was actually focused on Mars missions.
Okay.
And NASA wanted to know what were the biologic risks, the health risks to astronauts flying to Mars. And one of the chief ones is solar particle emissions. And the Earth, we've got our ozone, we've got our atmosphere that protects us from that. When you're in a little metal craft flying to Mars, you have no protection.
Yeah.
And if you get hit by an SPE, that can be a terminal event. You can receive enough radiation that it'll kill your bone marrow. So we really had to understand what we could do, ways to protect, what were the likely clinical outcomes of different amounts of SPE radiation while you were outside of Earth's protective atmosphere. So back then, the microbiome wasn't that well understood. We didn't study it.
Okay.
That's now, current for especially for astronauts on the space station. That's become incredibly important research. Um, there are so many physiologic changes being weightless for 6 months or a year causes that's fascinating.
Hmm. And while I have you, one other extremely interesting topic. You recently published on lipid nanoparticle composition drives mRNA delivery to the placenta, and again, a very obviously pregnancy, the placenta, everything that happens in those 9 months, a very unique microenvironment in its own right. And we talked a little bit previously about some of the especially the immune system, that the sort of the early embryo and the fetus doesn't have, I guess, certain amounts of different interferons and so forth to defend itself with, and has to rely on some other interesting mechanisms for protection against viruses and so forth. Say a couple words about what you're learning, both from a therapeutic perspective to how to address conditions per pregnancy, but also I guess some of the intricacies that are involved in studying that very unique microenvironment.
Yeah, so what people have to understand, we did these studies about 3 years ago when the
Mhm.
vaccine was first being developed.
Sure.
And what we wanted to know then was when you vaccinate a pregnant woman, is there a risk that the LNPs, the vaccine, goes to the placenta or goes to the fetus? We don't know if that's good or bad,
Mhm.
but we wanted to be sure that we understood what was happening. What we found and what that paper reported were very high doses of RNA LNPs. When you give a high dose, doses that you would use for gene therapy, you do see placental uptake. When you give a vaccine dose, 30 micrograms, 50 or 100 micrograms, we saw no placental uptake. We saw no effects on the fetus of mice at different stages. We spent a lot of time being sure that this new vaccine that we were giving had no effect on placentas, on fetuses,
Mhm.
on babies born. As it's turned out, it's one of its best uses has been in pregnant women,
Mhm.
because
Mhm.
pregnant women who get COVID have bad events happening to their babies. And that was prevented. First, Israel was the first to immunize pregnant women, but now it's common. We are studying the delivery of RNA LNPs to placenta into fetuses. We're looking at it as a therapeutic modality.
Right.
So nowadays, women have a variety of tests done while they're pregnant that tries to identify genetic defects, everything from immune deficiencies to cystic fibrosis, metabolic diseases. And the only approach now is sometimes you replace the blood when the baby is born and you continue to treat for the rest of the life. Sometimes you do bone marrow transplants. Our view is that if we can target the fetus at some stage of pregnancy, and it'll vary depending on the disease, we might be able to fix the genetic defects while the fetus is still in the mother.
Yep.
Just
Yep.
an injection of RNA LNPs. So it has enormous potential to change genetic diseases in our young. Someday we may no longer have cystic fibrosis. We may no longer have immune deficiencies. We may no longer have sickle cell and thalassemias and other diseases. We may no longer have phenylketonuria and other liver diseases. So we're looking at this technology as a way of treating fetuses.
Mm-hmm.
We know that vaccine doses do not go to the placenta, do not affect the fetus.
One of the topic I'd love to ask you about, and it's not directly related to mRNA, but still, thinking about health equity and thinking about sort of the themes of getting these technologies to the developing world. I know that you were also quite active in the whole area of microbicides and HIV, and have written a series of papers on sort of some of the different approaches, different compounds, and so forth that work in this. I learned all about semen amyloids from your papers. I didn't know that was a thing that existed, but talk about a little what's going on there, because clearly that's another area that has been worked on for a while, and it's had sort of its peaks and troughs along the way. But anything exciting happening in the area of HIV or other STD microbicides that we should know about, specifically as pertains to the developing world?
Yeah, I mean, it was a very interesting area of research. It's much less interesting nowadays because we've got things like PrEP,
Right.
pre-exposure prophylaxis with
Yep.
antiretrovirals, that when it's taken can be 90+% effective. We studied microbicides for a bunch of years, and we identified some interesting ones. The problem is always you have to take them before you have intercourse
Yeah.
for them to work. And often that's difficult. And it's especially difficult in many countries in Africa and other parts of the world where sex isn't always a choice for women. So ultimately, I think it's been determined that it's probably not the best approach to go since pre-exposure prophylaxis works so well and is so safe.
What what's coming up for 2024? Obviously, we covered a wide range of topics, everything from communicable, non-communicable, getting these technologies to lower income countries in the developing world. Anything that I missed as we enter 2024 that we should be aware of? And then if you could just say a couple words. I know you've written about your responsibility to get anti-debunking or debunking out there to the medical professionals and clear out some of this nonsense that we've been hearing about the last couple of years. But anything else you want to mention while we have you today? Please take the floor.
So to me, one of the most exciting things going forward is our in vivo gene therapy work.
Yeah.
take sickle cell anemia.
Mhm.
About 300,000 people a year are born with sickle cell. It's a horrible disease. It's a painful disease. Um, they're mainly in sub-Saharan Africa, but they're present through the entire world. The US just FDA approved a new sickle cell gene therapy
Yeah.
that takes out lots of bone marrow, infects it with a lentivirus, and gives it back. And it works well. It's going to cost $1 to $4 million a person.
Mm.
That doesn't work in sub-Saharan Africa. What we've been able to target bone marrow stem cells with very high efficiency, 60, 70% gene editing. We're developing gene editing technologies encoded as mRNAs that we can put into those targeted LNPs. And our ultimate idea is that we can go anywhere in the world with a freezer full of LNPs
Right.
and give people IV injections that will cure their sickle cell anemia. Once that happens, you can then expand that to all the other bone marrow genetic diseases, the thousands of immune deficiencies and other red cell, granulocyte, other deficiencies. They've already used RNA LNPs for gene therapy in the liver, and that's greatly expanding. That they've looks like they've cured transthyretin amyloidosis. We're also expanding that to PKU and to other liver genetic diseases. What this means is that not only does it make gene therapy easier and safer, one thing people don't mention is that when you take bone marrow out, you have to give chemotherapy to make room to give it back.
Yeah.
That makes people sick.
Yeah.
With in vivo gene therapy, you're targeting the stem cells that are in the body. They take it up and get gene edited and fixed. So it simplifies, it makes it much safer, it makes it much less expensive, and it allows worldwide use.
Yeah.
So to me, that's one of the other really fascinating and exciting directions that we're going in with this RNA LNP therapy. Now, you brought up a sore subject. As you can imagine, I get calls, emails, letters from a lot of ill people who threaten me, threaten my family, threaten my lab,
Yeah.
because there are websites that call Katie and I mass murderers. And all of this is based on social media nonsense,
Right.
where people make up things. They did a study. There were 13 physicians in the United States who accounted for about 70% of the social media anti-vaccine activity. When they looked at what these physicians did, many of them owned and sold homeopathic treatments for COVID. So I don't know if they believed in vaccines or not, but they believed in lining their pockets. And the way to do it was to say, vaccines don't work, they'll kill you, take my treatment. You then have even crazier people like surgeon generals at a few southern states who get this crazy idea about DNA inserting into chromosomes. What they don't understand is that the FDA has a set of rules for every vaccine.
Right.
And it says this is the amount of DNA you're allowed to have in the vaccine. Every vaccine has DNA in it. Protein subunits, inactivated viruses, live viruses, all of those things come from cells and have DNA in them. The DNA are tiny little pieces that can't possibly insert and have never been shown to insert into the chromosomes. RNA probably has less DNA than most subunit vaccines. But somebody has now got it in their mind that, oh, I can attack RNA vaccines and try to make them illegal because the FDA says there's DNA in them. And some crazy person said, oh, look, it can insert and cause cancer. It's never happened.
Well, I don't know what to do about some of that nonsense, but I think as you're doing, continuing to show the applicability and the efficacy and the safety of these products across infectious diseases, across non-communicable diseases, and ultimately, bringing an end to a lot of these unmet medical conditions. I think that's what you focus on. It's what you've been focusing on, and I think, I mean, it's just an amazing journey, Dr. Weissman, and I'm looking forward. And well, the fact that I'm sitting here in Philly, down the street from Penn, is extra exciting that it's all happening here. But really, wishing you and the team the best with all these projects as you continue to move them forward. Again, for everybody that's going to be listening to this particular episode of our show across the various podcast networks, or who will be watching on our YouTube channel. Again, you've been spending time with the amazing Dr. Drew Weissman, 2023 Nobel laureate in medicine, Roberts Family Professor of Vaccine Research, and Director of the Penn Institute for RNA Innovation. Dr. Weissman, I really want to thank you for taking all the time out of your schedule today to talk to us, to educate us for a little while. Obviously, thank you for everything you've been doing. And as we like to say here on our show, thanks for helping to create a better tomorrow for so many people out there via what you've been doing. It's a really great story.
Thank you very much.